Welcome to the first video of Neurogeneious, where we are going to be discussing all sorts of things on the intersection of neuroscience and genetics. My name is Rhea, and I am the founder of Neurogeneious, student at Phillips Exeter Academy, and a neurogenetics researcher. I'm super excited to see that you're starting this core neurogenetics course. So you might be wondering why this topic is so important to create an entire course on, but for me it's always been super thought-provoking because we still know so little about the human genome and the nervous system, even though these things are what define us as people and our interactions. It's not something like the mysteries of the ocean or the outer space, it's actually something within us. So the mere thought of it just being a mystery has always been sort of unsettling to me. So despite how important this is, there's only still one available neurogenetics course online, and that costs money. And this course specifically is dedicated to providing education on neuroscience and genetics and their intersection, which not only is super relevant to us as human beings, but the sequencing technology behind these fields could determine the future of healthcare and biomedical research. We'll talk more about these subjects in later videos, but the point I'm trying to make is that neurogenetics is something that needs to be emphasized more through accessible courses. So you might be wondering what exactly are the careers you could pursue in something as niche as neurogenetics, but contrary to common opinion, there are actually a ton of pathways you can take. One of the most known examples, not only pertaining to neurogenetics, but the biomedical field in general, is being a researcher in either industry or academia. This occupation can range from doing studies either as a postdoctoral researcher at a university or a research scientist at a more market-driven institution. There are also options beyond being a researcher, like working as an associate and eventually head professor at a university. But aside from researching or working in academia, there are tons of opportunities in pursuing neurogenetics in healthcare. There are many specific examples of careers here, but some of the main ones are being a genetic counselor on hereditary neurological disorders or doing neuropsychological testing for young children, and most examples here quite often involve pediatrics. Other careers that align with neurogenetics a ton are like working in bioinformatics or being a data analyst, along with working for pharmaceutical companies. We'll go more into these opportunities later, but it's important to know that there are a variety of tracks to take for pursuing something as specific as neurogenetics. Lastly, to end this introductory video, we want to provide some context on the development of this field before we really get into the nitty-gritty of the science behind it. This context is mainly provided from this article, which I would highly recommend reading if you want to extend your knowledge on this history. We're going to only be going over a brief summary, but I would say that there's definitely more nooks and crannies to it. So, just to provide some context, the correlation between hereditary traits and neurological diseases had been highly documented since actually the late 19th century, even before the idea and the study of genetics had really started to grow. However, the formal term of neurogenetics first emerged when American researcher Seymour Benzer discovered that single gene mutations could affect the behavior of Drosophila, which are essentially fruit flies. Previous signs of the intersection between genetics and neuroscience were seen in the discoveries of Friedreich's ataxia and Huntington's disease, both neurological diseases with very strong genetic linkage. After much discovery behind the structure of DNA and other common mutations in diseases, there is proof of a greater correlation between the nervous system and genetic information. Then, in the early 2000s, came the era of genetic modification, including sequencing technologies like whole exome sequencing, or WES, and whole genome sequencing, WGS, and these allowed us to identify single nucleotide variants or other insertion and deletion mutations. It may seem a little bit confusing now, but if you continue to watch these videos, I promise it won't only make complete sense, but it will also open your eyes to new mind-blowing, impactful concepts. So with that, I know that went by sort of fast and it was a lot of information, but I hope it was able to provide a nice overview of what we can learn in greater detail in this course.